Hello everyone, my name is Caden Vinovich. Today I will be presenting the research that I conducted these past 2 semesters under the guidance of my faculty mentor, Dr. Scott Grayburn. My research looked at the bacterial inhibition of Aspergillus nidulans fungus. I primarily worked with 2 different strains of Pseudomonas fluorescens bacteria, which Dr. Grayburn had isolated and named 18EI1 and 20EI1 respectively. Both these strains of bacteria have the ability to inhibit the mycelial growth and spore germination of Aspergillus nidulans. This fungus is closely related to fungi that can cause a human lung disease known as aspergillosis. My goal was to design experiments in which the bacteria and spores are grown together under different controllable conditions. Some of these variables were temperature, spore density, and the volumes of the spores and bacteria used. When grown together, the bacteria can create zones of inhibition, or clear zones in which the fungus could not grow. The conditions that promoted larger zones of inhibition were recorded, and further experiments were done to replicate these favorable results. The results garnered from this research can be used to contribute more data to understanding how to combat fungal diseases such as aspergillosis. So going down to the methods, there were 2 major experiments that I conducted these past 2 semesters. During the fall semester, I worked heavily with both 18 and 20EI1. These experiments focused on preparing plates with either the spores or the bacteria in top agarose. This top agarose contains the nutrients that both the spores and bacteria need to grow. If the spores are mixed together with the top agarose, then a small volume of bacteria is placed in the center of that plate. This bacteria then grows in a field of fungus and proceeds to inhibit the growth of the fungus around it in the growth media. On the flip side, if the bacteria was originally mixed with top agarose, then the spores would be spotted in the center of the plate to see the opposite of that original experiment I just described. So it's either the bacteria growing in the spores or the spores growing in the bacteria. The experiments that I conducted in the spring semester only involved 20EI1 and were focusing on growing the spores and the bacteria together before preparing them on the plate. So these are referred to as co-cultures because they're grown together prior to plating. These experiments looked at how allowing the bacteria to produce their antifungal compounds before being plated in the spores helped in inhibiting the fungal growth overall. So if we go to the middle of the poster here, the table shows measured clear zones by both 18 and 20EI1. Most of these experiments were done where the plates were left to grow at 30 degrees Celsius. Figure 3, which is the small picture on the right, shows a plate of 20EI1 with a zone of inhibition in the middle of the plate. This plate had spores in the top agarose and a spot of the 20EI1 bacteria. You can see underneath the 6, that black circle in the middle is where the bacteria was originally spotted and grew. That zone of discoloration is the zone of inhibition that extends from that bacterial growth, and then that is what is measured as a result and as that zone of inhibition. So the bottom larger picture shows 2 plates from the co-culture experiments. The plate on the left exhibits what we classified as complete inhibition, where the spores that were originally spotted on the plate were unable to germinate in the growth media with a co-culture of 20EI1 and spores. So it couldn't grow at all because of the co-culture in the growth media. So the plate to the right of it is under the exact same conditions, except that instead of there being a co-culture in the top agarose, there was only the 20EI1 bacteria. So this showed that the production of inhibitory compounds before being prepared on the plate leads to significant inhibition compared to no co-culture. And so this leads me into my overall conclusions from my research. So Aspergillus nidulans grew much better at 30 degrees Celsius than 24 degrees Celsius. We found that the fungus has multiple stages of growth and that 18EI1 and 20EI1 were able to inhibit those stages. Complete inhibition of the fungus was only ever recorded in plates where the bacteria was in the growth media to start, or the co-culture for that matter. So overall, this research still has a long way to go in terms of data collection. During my time in Dr. Grayburn's lab, I was able to learn the ins and outs of laboratory work that I have never had the opportunity to experience before, and for that I am extremely grateful. I want to thank you all for your time, for listening, and I know this isn't an open forum, but if you have any questions or if you go to the in-person cure event, then I would be more than happy to answer them there. Thanks.